Today we will be discussing additive manufacturing for biomedical applications. This presentation was prepared by group M15.
I'm Nada Nasr.
I'm Andrew Nosworthy.
And I'm Emily O'Day. Additive manufacturing, also known as 3D printing, is defined as a subset of rapid prototyping that produces prototypes or fully developed parts built using a layer-by-layer approach as opposed to traditional subtractive techniques. The first patented additive manufacturing technology was stereolithography, created by American inventor Charles Hall in 1986. This creation led to the widespread development of a number of 3D printing technologies, including fused deposition modeling, selective laser sintering, and laminated object modeling, to name a few. From its origins as a means of rapid prototyping in the mid-1980s, additive manufacturing has had countless applications, and as technologies advance and diversify, so do the possibilities for their use. In terms of biomedical applications, additive manufacturing technology has many possibilities, such as dental aids and implants, bioprinting, and pharmacoprinting, which we will discuss today. From automotive and aerospace to intricate personalized biomedical implementations, additive manufacturing has come a long way and has a bright future.
Technology generation of dental applications. Daily studies are conducted on dental applications and how they can be improved. Dentures, bridges, and crowns are an example of mouth restorations that are able to be 3D printed. The most common material used to 3D print these implants is a titanium alloy, specifically known as titanium, 6 aluminum, 4 vanadium. The fabrication of these restorations is done through a process known as direct metal sintering. In DMS, an ultraviolet curable photopolymer resin paired with an ultraviolet laser is used to cure the liquid into a solid object. Following the fabrication of these implants are one or more small surface modifications. Some of these modifications are as follows: sandblasting, acid etching, anodization, calcium phosphate crystal deposition, and chemical modification. One or more of these steps are required to achieve the desired surface finish. Tixos is an example of a company specializing in the production of dental implants using additive manufacturing. This company utilizes direct metal sintering to fabricate their implants. Tixos uses an EOSint M270 ytterbium fiber laser system. An example of this machine can be seen in the image to the right. The printers used at Tixos have a building capacity of 250 by 250 by 215 millimeters cubed. Perfect for producing partial dentures, crowns, and bridges for the mouth. After printing, Tixos follows a standard cleaning procedure of multiple cycles of bathing. Starting with a 5-minute sonication in distilled water, the cleaning is followed by immersion in a sodium hydroxide and hydrogen peroxide solution. This cycle is repeated twice, and then the implant is cleaned with distilled water for another 5 minutes.
The current bioprinting methods in use consist of droplet-based, laser-based, and micro-extrusion bioprinting. These technologies use biomaterials such as living cells, DNA, and biochemicals to facilitate cell growth to create scaffolds within which the cells can proliferate and form tissues. Currently, there are a number of both natural and synthetic biomaterials used in bioprinting. 3 of the most common applications of bioprinting today are modeling for preoperative planning, organ and tissue printing, and drug and cosmetic testing. Although resin models used in preoperative planning do not use biomaterials, they have become an incredibly useful tool in decreasing surgical time and consequently decreasing the risk of surgical complications. Structures such as bone fractures and tumors can be scanned and modeled exactly as they appear in a patient. This allows for medical professionals to increase the accuracy of things like incision and screw placement and tumor excision while decreasing risk to surrounding tissues. Currently, organ and tissue bioprinting produces scaffolds to replace mainly bone or cartilage that has been damaged or destroyed. The parts produced can have very specific porosity, allowing for the scaffolds to be completely bioreabsorbed by the body. Finally, drugs and cosmetics can be tested on bioprinted tissues, decreasing the need for animal testing, which is both highly controversial and not completely accurate for human comparison. Pathological organs and systems can also be created to test long-term treatments for afflictions such as cancer and chronic illnesses.
Pharmacoprinting describes the process of additive manufacturing procedures to fabricate printed pharmaceuticals. There is currently one available 3D printed drug called Spertom, which is an anti-epileptic drug. Spertom is made using zip dose technology, which is a type of 3D printing that prints layer by layer of powdered medication that gets stitched together by droplets of liquid, causing microscopic bonds forming a matrix structure that forms a pill as shown in the bottom photo. This process creates a highly porous pill that can instantly dissolve, making it possible to administer medication on a seizing person without it being a choking hazard. The figure shows a visual of the described process that Zipdose technology uses to create Sprotom.
Additive manufacturing in the future of biomedical. The traditional way of creating any specific restoration for a human mouth was through the use of a mold. This mold is treated as a workspace for the dentist, acting as the ideal canvas for the design of any implant. Though this traditional manner of fabrication is a very timely, tedious, and inefficient mode of production. With 3D printing, theoretically any part of the mouth can be created, eliminating the need for the model workspace. Companies are exploring more applications for DMS, and currently there are several studies in multi-material hyperrealistic polymer dental model printing, also known as real teeth.
One of the biggest challenges currently facing the industry is the state of biomaterials in terms of vascularization of bioprinted tissues. It is expected that advances will be made regarding ways to ensure the porosity is correct and maintained as the tissue develops to provide the necessary space for vascularization. This will greatly extend the time available for testing and use between printing and cell death of the biomaterials used. Increases in cell type variation used in bioprinting could decrease heterogeneity and data variability in the results of testing and research, leading to more concrete and accurate conclusions. Some other predicted advances in biotechnology include the integration of systems to allow tissues to be incubated and matured immediately following printing in a closed environment. This could potentially lead to more rapid clinical testing outcomes. Additionally, the integration of biomaterials with microfluids is expected to facilitate research in certain pathologies within the microenvironments created. Another expected area of development is the development of high-throughput manufacturing of 3D architectures, which describes the automation of processes enabling higher rates of repetition and production, easing repeated testing, and leading to larger sets of resultant data. These are all expected to increase the applications of the discussed technologies. These are just some of the foreseeable advancements in bioprinting technologies expected to be developed within the next decade. There are undoubtedly countless other possibilities that have not yet been reached by researchers, medical, and engineering professionals. The growth so far in the bioprinting industry has been incredibly fast and influential both in research and in direct implantation, and it seems this growth will continue, if not increase, in the years to come.
There was difficulties approving Sprotom since the FDA was not familiar with pharmacoprinting technologies. But now that one drug has been approved, possibilities for more 3D printed drugs are endless. For instance, in the next 5 to 10 years, it could be possible to have pediatric hospitals 3D print starfish-shaped pills for terminally ill children. In terms of other possibilities for pharmacoprinting, Sprotom was made using Zipdose, which relies on the drug being of a powdery nature. However, with inkjet technology, it could be possible to fabricate drugs that are liquid-based by initiating chemical reactions in the printing chamber, or by using vapor deposition to create many more compounds, some of which are not soluble enough to work with current drug production methods. The challenge, of course, will be to find a way to digitalize all the chemicals needed for a drug so that it can all be built from scratch. Pharmacists will have to be trained and adapted, since drug productions can start happening in their pharmacies. Lastly, with this new individualized therapy approach to medicine, liability might become a problem that the FDA will have to reform and accommodate. When an adverse reaction occurs, liability will lie in a gray area where the printer is considered a medical device, the ingredients or the base materials are pharmaceuticals, and the person manufacturing the drug might also be the consumer.
Additive manufacturing for biomedical applications has helped enhance the medical industry to meet patients' needs. There have been major innovations in dentistry, bioprinting, and pharmacoprinting. With the speed and low cost of additive manufacturing, it will soon be possible to meet patients' needs on the spot, as well as create more medically needed equipment and drugs that cannot be produced and tested using traditional methods. There might be a future where a 3D printer can print a medicine and an organ, test their interactions, and monitor it. The future of biomedical is additive manufacturing.